All right, so today's topic is 8.2, which is all about cellular respiration. And so before we deep dive into that, let's go ahead and do a little bit of review from topic 1, where we were looking at cell structure. So we know that the eukaryotic cell, that the hallmarks of this cell type is the presence of these membrane-bound organelles, which include things like the nucleus, where the DNA is stored. That's, where your linear multiple chromosomes hang out in the cell, and that is immediately adjacent to the rough endoplasmic reticulum, which along with the Golgi apparatus is responsible for the generation and secretion of proteins that are either going to the membrane or outside the cell. And then we also have the mitochondria over here, which is colloquially known as the powerhouse of the cell, but more generally, or more specifically in our context, is the site of aerobic respiration. And so just to remind that aerobic respiration is the type of respiration where oxygen is required to break down glucose to generate that large amount of 36 ATP. And so while we're talking about the mitochondria, let's go ahead and also review endosymbiosis, right? So this is the theory of how both mitochondria and chloroplasts came into existence. So this is also from topic 1. And so this is where one prokaryote engulfs another. And instead of that smaller prokaryote being digested, it formed a symbiotic relationship with its larger neighbor here, and that this is what led to the presence of mitochondria and chloroplasts in the higher order cells. So this is where eukaryotes originated from. And so just to review what some of the evidence was for endosymbiosis is that if we compare the structures of a free living prokaryote with both mitochondria and chloroplasts, all three of them have that single circular chromosome. All three have those smaller 70S ribosomes, which are going to generate protein. All three have a double membrane. In the case of the prokaryote, that would be the cell wall versus the plasma membrane. And all three divide by binary fission, which is a form of replication that is different than the cell cycle in eukaryotic cells. And then just a reminder that prokaryotes do have some structures that are present only in the prokaryote, like pili, those attachments. Since mitochondria and chloroplasts are not free living, they don't have any reason to attach. But what they do have in common, those single circular chromosomes, the 70S ribosomes, the double membrane, and that dividing by binary fission are all evidences for endosymbiosis. So coming back to our current topic, right, that mitochondria is the site of aerobic respiration. And so this is the type of respiration that does require oxygen, that is happening inside the mitochondria itself. And so this is the breakdown of glucose, which is going to produce that 36 ATP. And then as waste products, you're going to have carbon dioxide and water. And so in previous talks, when we were in topic 2.8, we talked about respiration as if it was just one reaction. And actually, there are 3 separate steps to this. So first of all, aerobic respiration is preceded by a reaction called glycolysis. So I think I've mentioned this in previous lectures, but now we're going to be really explicit that aerobic respiration itself is preceded by glycolysis, which is an anaerobic type of respiration that's going to generate ATP. And then that is going to occur in the cytosol. And so that has to occur before that pyruvate can enter even aerobic respiration, which itself is broken into 3 components. The link reaction, which is occurring in the interior of the mitochondria called the matrix. And then that is followed by something called the Krebs cycle, which is also occurring in the matrix of the mitochondria, which is then followed by the final step, or final cycle, whatever you want to call it, in aerobic respiration, which is the electron transport train, which is occurring in the folds of the inner membrane. So where we've been referring to aerobic respiration as just one thing, it is really 3 separate reaction cycles, and again, preceded by glycolysis. And so we are going to be focusing a lot on where these different reactions are occurring, so I just want to give you some landmarks about the structure of the mitochondria. So, like a prokaryote, it's got an inner and outer wall. So this one has an outer membrane and an inner membrane. It's got the same 70S ribosomes that you would see in a prokaryote. It's got its own circular chromosomes that you would also see in a prokaryote. But what is slightly different is that instead of calling this like the plasma membrane, we're calling this the inner membrane. And what is also different in a mitochondria is that this inner membrane does not cleave close to that outer membrane. Instead, it folds. And it folds into these structures called cristae. And so these exist in a mitochondria to increase the surface area. Because later, when we're looking at the electron transport chain, those reactions are happening within the membrane of the mitochondria, this inner membrane. And so to pack more of that into the mitochondria, the folds are there in the cristae to increase that surface area to allow more of those reactions to occur. So within the inner membrane, that's where we're going to have that part 3, the electron transport chain, which is occurring within the cristae in that inner membrane. You'll hear it referred to as other names like oxidative phosphorylation, that it's generating the proton gradient for something called chemiosmosis. We'll go into more detail of what that is later. And then we have the matrix, which is this interior space inside the inner membrane. And that's where our link reaction and Krebs cycle is going to occur. And then we also have the intermembrane space, which is where the proton gradient that is created by the electron transport chain, those protons are going to accumulate in this intermembrane space. So I'm just giving you landmarks of what you're going to see and where these different reactions occur up front. Okay, so overall, the overall reaction of aerobic respiration is glucose being broken down into these 36 ATP. And then as a waste product, we are generating carbon dioxide, which we breathe out, and water vapor, which we also breathe out. And we know that as humans, right, we need oxygen to breathe. And so what we're going to see is that where exactly all these different components fit in to this cycle, and where these different waste products are being created. So as I said, aerobic respiration itself is preceded by glycolysis, which is an anaerobic form of respiration that is occurring in the cytosol, right? So we're not in the mitochondria at this point for glycolysis, we are out here in the cytosol. So in glycolysis, what is happening is that glucose is being broken down into 2 pyruvates and a small amount of ATP, which is a form of energy. And we've kind of glossed over what the stuff is that's happening during this breakdown of glucose into pyruvate. And so what we're going to look at is some of the things that are happening during the breakdown of glucose into pyruvate. And so again, this is occurring in the cytosol, not in the mitochondria. So this is the entire process of glycolysis. You do not need to know the whole steps. What I want to focus on is the kinds of reactions that are occurring in here. So one of them is something called phosphorylation. And phosphorylation is where you're moving a phosphate from one molecule to another, and that phosphate is frequently taken from ATP itself. And so there's a phosphate that's moving from ATP to these different molecules. And you can see that this occurs in a number of places throughout this reaction. So just to focus on that type of reaction again, let's look at ATP, which I've previously referred to as energy bucks, who is a frequent donor of a phosphate to other molecules. So adenosine triphosphate is the full name of ATP, right? Because it's an adenine base attached to a ribose sugar attached to a phosphate with 1, 2, 3 phosphates. And if you just kind of covered this part and you're thinking, hey, this looks a lot like a nucleotide, you would be correct that this is a phosphate attached to a ribose sugar. And I know it's ribose because it's got 2 hydroxyl groups here attached to an adenine base. This is, in fact, a nucleotide by itself. So this doubles as both a participant in like the RNA and DNA structures, but also ATP is used as an energy source and as a source of phosphate. So this is called adenosine triphosphate, as I said, because it has 1, 2, 3 phosphates. And what is important to know about ATP is that this bond right here between these two phosphates is highly unstable and it carries a lot of energy in it. And you can think of it as like sort of like a loaded spring that this phosphate wants to pop off. And when it does, it's going to donate energy to a process. So we can do a number of things with that. So not only can we get some energy when we break this bond, but we've also now freed a phosphate that can be attached to another molecule. And so when this happens, ATP, instead of being adenosine triphosphate, is converted to ADP, or adenosine diphosphate, because now it only has 2 with this free phosphate that is usually transferred to another molecule. So this movement of phosphates to another molecule can be used for lots of reasons, not the least of which is like signaling, or to, again, create another unstable bond somewhere else so that we can use that energy to facilitate a different chemical reaction. So ATP, right? Unstable bond. I use this as a donor of phosphates for a lot of my reactions, right? And so that movement of a phosphate to some other molecule is called a phosphorylation. And so if we look at different kinds of reactions that you're going to see, I can phosphorylate pretty much really anything. So if glucose, I can put a phosphate group that, again, is donated from that ATP. So this phosphate is going to then come off ATP to create ADP, resulting in this glucose 6-phosphate. So the phosphate that was on ATP is just going to end up somewhere else. And so again, this is what a phosphorylation reaction looks like. And so when these phosphates are moving around, that's the kind of reaction that you're going to see. And so if you look at glycolysis, you see this pattern of where I'm taking a phosphate group from ATP and I'm putting it on some molecule. And so this phosphorylations are happening really all over glycolysis, and I can even do it in the reverse direction where ADP is phosphorylated to create ATP. So point being, phosphorylation reactions are occurring in glycolysis. There's a second type of important reaction here that we'll talk about in a second. So phosphorylation is occurring in glycolysis. So now let's look at the creation of this NADH and 2 ATPs. So if I look at the whole of glycolysis, I see this type of reaction frequently where I have an NAD plus being converted to an NADH. And so this kind of reaction is called a reduction reaction. And it's weird to call it a reduction reaction because I always thought, well, it's gaining a hydrogen. Why is it called reduction? And that's because really what they're following is the electron that is hanging out on hydrogen itself. So when NAD plus is being converted to NADH, it is because it is reducing the charge of that molecule and going from a positive to a neutral because I added an electron. So I reduced the charge. So I know it's backwards. I'm sorry. So we call these reducing reactions because it's reducing the charge when I go from an NAD plus to an NADH. And so the other version of this is where you'll see a FAD molecule going from FAD plus to FADH2. They're being reduced because I'm reducing the charge by adding an electron on a hydrogen. And so this hydrogen with the electron doesn't come from outer space. Something else was losing that hydrogen with an electron on it. And so whatever is donating that hydrogen and electron is being oxidized because it is losing an electron. So reduce, I gain a hydrogen, oxidize, I lose it. Okay, so this right here is a reduction reaction. It's the only one that we see in glycolysis, right? So just to review that a little bit, glycolysis is happening in the cytosol. And I'm going from glucose, and it is being broken down into pyruvate. So this is the anaerobic part of respiration. It is occurring in the cytosol. The main products are the pyruvate with a little bit of ATP. And so this pyruvate is now going to move into the matrix of the mitochondria and go into something called the link reaction. And so some things are going to happen to this pyruvate, and we'll talk more about what that is. So the whole point of the link reaction is to take in pyruvate, and it's basically going to produce this other molecule called acetyl-CoA. So glycolysis, which is anaerobic, has already happened, right? So glucose goes to pyruvate. It produces a small amount of ATP. Glycolysis itself contains reactions where those phosphorylations of different molecules is happening. That reduction of NAD to NADH is occurring within glycolysis. In the link reaction, right, we start with the pyruvate, which was created in glycolysis, and we're moving into the matrix of the mitochondria. So it's this interior portion, this interior space of the mitochondria where the link reaction is occurring. And the goal of the link reaction is going to be to produce a molecule called acetyl-CoA. So to get from pyruvate to acetyl-CoA, a couple things have to happen. So one of them is that this carboxyl group on the pyruvate is going to have to go away. And so when a carbon dioxide is kicked off, we call that a carboxylation, or a decarboxylation, sorry. So this pyruvate, or this carboxyl group on the pyruvate, is going to be freed from the pyruvate, and so we call that a decarboxylation. And like every, action has an equal and opposite reaction. If I gain a COH, that's a carboxylation. When I lose it, it's a decarboxylation. So anyway, I'm back over here. Pyruvate comes in from glycolysis. I undergo a decarboxylation. At the same time, pyruvate is going to be oxidized. It's going to donate a hydrogen to this NAD plus to create NADH. So this NAD is reduced to become NADH. The pyruvate is going to be the donor of that hydrogen. So pyruvate is oxidized, NAD plus is reduced to become NADH. And the end result is that acetyl-CoA is the end product of that link reaction. So within the link reaction, we have both a decarboxylation and an oxidation reduction reaction. So pyruvate is decarboxylated, right? We lose the carbon dioxide. And pyruvate is oxidized to reduce that NAD plus to NADH. And this is all occurring in the matrix of the mitochondria, and the outcome of this reaction is the product acetyl-CoA. So here we are, just again from the top. We start with glycolysis, which is anaerobic and occurring in the cytosol, which breaks down glucose into pyruvate with some of that NADH. That pyruvate goes into the matrix for the link reaction, which decarboxylates that pyruvate and oxidizes it to reduce the NAD+, thus creating acetyl-CoA. So once we have acetyl-CoA, we're ready to enter the second portion of aerobic respiration, which is the Krebs cycle. So this can also be known as the citric acid cycle. It goes by both names. So the Krebs cycle, like the link reaction, is occurring within the matrix of the mitochondria. So we're still in here, we're still in this interior part. And so the acetyl-CoA was already created in this space by the link reaction. And what's going to happen is that acetyl-CoA is going to go through a series of decarboxylations and oxidation reactions. And the point of the Krebs cycle is the creation of electron carriers, NADH and FADH2. And so to get these are reduction reactions that are going to go from that NAD plus and FAD plus to the NADH and FADH2. So these are reduction reactions. And then these electron carriers are then going to go on to the electron transport chain, which is our third reaction in respiration. So that's the overall of the Krebs cycle. So if we look at this another way, right, we have our pyruvate coming in. Again, that's decarboxylated here. That pyruvate is oxidized to reduce NAD plus to NADH to result in that acetyl-CoA. So that was a link reaction. And so acetyl-CoA is going to go through, again, that series of reactions where decarboxylations are happening, reductions are happening, because again, the main point of the Krebs cycle is the reduction, of these electron carriers. That NAD plus is reduced multiple times to create an NADH, and that FAD is reduced to create FADH2. And so what these are doing, these reduction reactions are doing, are putting electrons onto these molecules. They're reducing them. And so these are the important products that are going to go off and feed into the electron transport chain. So that's one thing that's happening within the Krebs cycle. The other thing that's happening is some decarboxylation events. So that CO2 that we breathe out, this is one of the places that it comes from. So like this 1, 2, 3, that CO2 that we breathe out as a byproduct of breathing, that is the source of it, right, when we're breathing out. So this decarboxylation is happening to these groups, these acyl groups that are on these molecules as they are going through the Krebs cycle. So you don't need to know the intermediary steps like citric acid, oxoallyl acetate. That's outside the purview of this course, but what you do need to know is how many carbons are present in each step. So acetyl-CoA, as you saw before, has 2 carbons. And at the beginning of the Krebs cycle, acetyl-CoA combines with oxoallyl acetate, which is 4 carbons, and becomes 6 carbons. So at the start of the Krebs cycle, we have a 6 carbon chain in citric acid. And so if I have a 6 carbon chain and I lose a carbon dioxide, well, I've subtracted 1 carbon and now I'm at 5 carbons. And if I have a decarboxylation event again, which I do, I go from 5 carbons to 4 carbons. And so for the rest of the steps, there are no more decarboxylations, so I stay 4, 4, 4 all the way around until acetyl-CoA with its 2 carbons again combines with oxoallyl acetate to make that 6 carbon citric acid, and the cycle starts again. So what you need to recognize here is the types of reactions that are happening, that the conversion of NAD plus to NADH and of FAD plus to FADH2, that these are reduction reactions, that decarboxylations are occurring at these 2 steps here, and that the number of carbons starts at 6, and after the first carboxylation goes to 5, then after another decarboxylation goes to 4 and stays at 4 until the cycle starts again. So that's what you're held to as far as the Krebs cycle is occurring, right? And so just to revamp where we've been, in glycolysis, that anaerobic portion, which is occurring in the cytosol, glucose is broken down into pyruvate, which then feeds into the link reaction in the matrix. So that pyruvate is decarboxylated and oxidized to reduce NAD plus to NADH, resulting in acetyl-CoA. That acetyl-CoA is going to go into the Krebs cycle, really creating these reduced molecules of NADH and FADH2, which are going to feed into our final portion of aerobic respiration known as the electron transport chain. So you will see lots of variants of representations of this. I'm just going to take you through some of these. So Krebs cycle, again, occurring in the matrix, creating these reduced molecules NADH and FADH2 that are going to feed into the electron transport chain, and those proteins are embedded in that inner membrane. So they're embedded within those cristae. So this is just another representation of the same thing, that we have the interior of the mitochondria, which is the matrix, which is where the TCA cycle, the Krebs cycle, is occurring. So it's the same name. And we are creating these reduced molecules of NADH and fat H2 to feed into the electron transport chain, which is embedded in that inner membrane. So just a third representation. So again, citric acid cycle, Krebs cycle, it's the same thing. The link reaction, which starts with pyruvate, goes to acetyl-CoA, feeds into the Krebs cycle, creating these electron carriers, these reduced forms of electron carriers, NADH, which is going to feed into the electron transport chain. So to focus on that, right? So now we're down here. We've talked about glycolysis. We've talked about the link reaction, which happens in the matrix of the mitochondria, which is then followed by the Krebs cycle. And so now we're on the last part, which is the electron transport chain. So the complexes of the electron transport chain are numbered 1 through 4. So 1, 2, 3, 4. And ATP synthase is usually called complex 5. But ATP synthase is really also the star of the electron transport chain. And so these are embedded within the inner membrane. And what is going to come into this are the products of that Krebs cycle. So these are our electron carriers, the NADH and fat H2. So these were reduced in the Krebs cycle, right? That NAD plus and fat plus were reduced to create NADH and fat H2. And so what is going to happen is the reverse reaction. So NAD plus was reduced to create NADH. Well, now it's going to be oxidized. So the hydrogen that was on this NAD plus is going to be oxidized. It's going to be donated to this complex 1. And the fat H2 is also going to be oxidized. It's going to have those hydrogens taken away from it, and their electrons that were on there are going to leave this molecule and go into the complexes. So there's a couple of things that happen once these molecules interact with complexes 1 and 2. So first of all, the hydrogens that were carrying the electrons are taken off. So these electrons are then passed down the chain through complexes 1, 2, 3, and 4. So this is being facilitated by other molecules that are not part of this diagram. They're outside the purview of this class. So the electrons are on the hydrogens. They get into these complexes, and then they're ferried down to complex 4. At the same time, once those electrons are removed from the hydrogens, now it's a proton. And now that proton is going to be pumped out into the intermembrane state. So the hydrogens are separated from their electrons, creating protons, which then are pumped out. So these things are happening at the same time. So what is occurring is that we are creating a proton gradient here. And so the more protons I have on the outside, right, as soon as I open a channel, they're going to want to run down back, with their concentration gradient. And we're going to use this proton gradient to power ATP synthase. So over time, after the NADH and fat H2 are oxidized, I'm going to have a buildup of a proton gradient in the intermembrane state. But remember, I had these electrons floating around. And so those electrons, go back. So those electrons are accepted by an oxygen. So we need oxygen to breathe because of this chemistry that's happening right here. So you cannot make ATP without oxygen being a final electron acceptor. So those electrons that were being passed down must encounter oxygen. And then this is going to create water, which is also why you breathe out water vapor when you're breathing. So we have two things happening here. So once I do that, I've put my electrons onto oxygen, which is that final oxygen electron receptor. Now I have this really great proton gradient. So what is going to happen is that once the proton gradient is created and I open a channel, which is happening in ATP synthase, that's going to cause the rotation of the molecular motor down here. So once I open that channel and that proton gradient is driving the activity of ATP synthase. So as the name implies, ATP synthase is synthesizing ATP, and it's doing that by taking in ADP and phosphorylating it back to ATP. So every rotation that we have occurring with the movement of these protons is going to result in the spinning of these propellers in ATP synthase. And so that is going to allow phosphorylation of ADP to occur. And that's where you get these massive amounts of ATP energy from the mitochondria, right? So there's a couple of things to note here. The use, so the creation and the use of this protein gradient is known as chemiosmosis, or at least it is in IB land, right? Where we go through all of this, right, to make the proton gradient to power the function of ATP synthase to make that ATP. And because we're coupling the oxidation of these electron carriers, so that NADH, that fat H2, we're oxidizing them, right? We're taking away their electrons that were hitchhiking on those hydrogens. Because we're oxidizing them and we're using this chemiosmosis to drive the activity of ATP synthase, which is effectively phosphorylating in ADP to create ATP. That's why this process is often referred to as oxidative phosphorylation, because I'm using the oxidation of these electron carriers to create this proton gradient, which is driving the activity of this complex to phosphorylate ADP. So it's oxidative phosphorylation. I've got oxidation reactions happening to facilitate the phosphorylation of that ATP. So this is just another representation of the process, a prettier one, that you have these electron carriers that would be like the NADH, and that the electrons are skipping up these complexes, being carried to the neighboring complexes down the way. That's what the little lights are. So this would be NADH being oxidized. This would be fat H2 being oxidized. So the electrons are being carried down the way, all the way to complex number 4. And so those electrons, again, are going to end up with our oxygen molecules. That's what these guys here, I think, are referring to. So we're going to follow our little electrons here. And so they will couple with oxygen to create water. Yeah, so these are oxygen molecules that are coming in. Those oxygens are accepting those electrons, and then it's being spit out as water into the intermembrane space. And what is also happening, so these other little balls that are here are the hydrogens, right? The protons, to create that proton gradient, to allow that chemiosmosis to happen. And so this is just a view of ATP synthase, which is using the proton gradient to rotate and thus create ATP from ADP. So as that's going on in there. All right, so to recap, we start with glycolysis, which is occurring in the cytosol. So this is an anaerobic process, does not require oxygen to occur. And we're going from glucose, we're producing pyruvate and a small amount of ATP. So we have some phosphorylation reactions happening in there. We have a reduction reaction happening in there. That's how we get this NADH. But the main player of glycolysis is pyruvate, which is then going to go into the matrix of the link reaction. So that pyruvate is decarboxylated. It's also oxidized to reduce NAD plus to create NADH. And the main product of the link reaction is acetyl-CoA, which is then going to go into the Krebs cycle, where we go through those reduction reactions to create NADH and fat H2. These are my electron carriers, which are going to go into the electron transport chain. So these NADH and fat H2, they're donating their electrons on the hydrogens. The electrons go down through complex 1 through 4, where oxygen is the final electron acceptor, and then it creates water. And then at the same time, the hydrogens themselves are used to create a proton gradient, which allows chemiosmosis to power that ATP ATP synthase, allowing the phosphorylation of ADP to ATP. And so that is how we end up with 36 ATPs from aerobic respiration. And that's all, folks.